Hello, I am Eric Jans, one of the two primary researchers on the tick and tick-borne pathogen surveillance as a public health tool updating Kansas geographic distribution maps research done by the Pittsburg State University Department of Biology with our professor Dr. Anurada Ghosh in collaboration with Aaron Petro from the Bureau of Epidemiology and Public Health Informatics in the Kansas Department of Health and Environment. Some background information on ticks, the focus of our research. Ticks act as a vector for a multitude of diseases relevant to public health, and they do this through the transmission of viruses, bacteria, protozoa, and helminths. So being a vector that is able to transmit diseases through all four of those is unique to ticks. Diseases with thoroughly researched symptoms, such as Lyme disease and anaplasmosis, which are transferred by Ixodes scapularis, or the deer tick, ehrlichiosis, the disease transferred by Amblyomma americanum, which you can see here labeled as the Lone Star tick, and spotted fever rickettsiosis, the disease transmitted by Dermacentor variabilis, which you can see labeled as the American dog tick, all of which result from tick bites. Greater surveillance efforts identifying the prevalence of these specific tick species, as well as the variation in prevalence due to environmental factors in the Great Plains area, is needed, as research on this topic has so far been fairly underdeveloped. The research in southeastern Kansas being limited has resulted in the data on reported and established tick populations in Kansas counties being underrepresented compared to the reports of the actual number of diseases occurring in this area. The sites that we went to collect ticks and survey for the ticks were two spots in Crawford County, which is the county that Pittsburg State University resides in, as well as two location or collection sites in Anderson County, one of the other counties nearby Pittsburg State University. In Crawford County, Wilderness Park, which is the small rectangle in Figure 1, and Mindland, the area in the bigger rectangle in Figure 1, were the two sites in Crawford County visited. We visited both sites once a month between March and August in 2023. And as you can see in Figure 1A, the transect where we collected ticks for Wilderness Park. Figure 1B, the transect collected from Mindland, and the second transect from Mindland, Figure 1C. All three of these transects representing a 750 square meter area. And then in Anderson County, the two collection sites were Lake Garnet Park, as you can see in the top right rectangle, and Cedar Valley Reservoir, which occurred in Garnett, Kansas, both of which occurred in Anderson County. The Cedar Valley Reservoir was more of a near water collection site, while the Lake Garnett Park was more of a camping ground, and these two sites were visited a total of 3 times during the months between May and August of 2023. All of the collection sites in both counties had frequent visitors, hikers, bikers, and campers. Ticks were collected using the flag drag technique along the side of walking trails and gravel roads. As you can see in the right picture, the flag and drag technique involved taking a white linen cloth about 1 meter by 1 meter in length, dragging along the side of trails, and then every so many steps laying down on the ground to examine for ticks. If ticks were spotted, we collected them in vials to be later counted and frozen. The collected ticks, when counted, were sorted by species, sex, and life stage at our lab. And as you can see, Wilderness Park is the location of where these pictures were taken, and that was the first collection site in Crawford County. The data that we collected, as you can see here, we collected a total of 495 adult and nymph ticks. The majority of ticks collected were identified as Amblyomma americanum ticks, or the Lone Star ticks, which accounted for 90.3% of our total collected ticks. We found of the Amblyomma americanum ticks, 203 were male, 170 female, and the remaining 74 were nymph, which means that they had not gotten to the stage of their life cycle where sex had been determined. The second most common tick that we found were Dermacentor variabilis ticks, which accounted for 9.7% of the total ticks collected, which accounted with 25 males, 23 females, and the remaining 42 were nymphs. And some interesting or significant parts of the data were that a total of 12 ticks were collected from Anderson County, 10 Amblyomma americanum ticks, and 2 Dermacentor variabilis ticks. Temperature was higher and humidity was lower in this county on collection days compared to Crawford County. On our July through August visits at Crawford County, which you can see in the last column on both the Wilderness Park and Mineland area, the only ticks found were really hundreds of larvae, and therefore they were not identified because of their smaller size and lack of development in the life cycle. On our last visit to Anderson County in August, numerous larvae ticks were also collected. None of the collected ticks were Ixodes scapularis, which is the primary vector of Lyme disease. As for our temperature and humidity data, the temperature and humidity were higher for the later months of the summer than the earlier months of the summer at Wilderness Park, and fairly consistent for our trips to Mineland. The total amount of ticks found in our about once a month visits were highest during the months of May and June for both Wilderness Park and Mineland. As a result of our research, we were able to give to the Kansas Department of Health and Environment our data to update the Kansas tick geographic distribution maps. So as you can see here, we have the cropped pictures of Kansas. The reason they are cropped is because counties left of the cropping have no reported data. The blue labeled counties have established populations of ticks as of 2022, which means they have 6 or more ticks of a single species, or more than one life stage of a single species. So on the left you have the Amblyomma americanum distribution map, and on the right you have the Dermacentor variabilis distribution map. And then in the yellow highlighted counties is representing a reported tick population, which means that there were less than 6 ticks of a single life stage collected in the county within the same 12-month period. However, there were ticks found. As a result of our research, Anderson County went from being a reported to established population of Amblyomma americanum ticks, and then Crawford County went from being no reported data or no record to an established population of Dermacentor variabilis ticks. So that is the blue rectangles. As a result of the research, we hope to in future years, go to the red circled counties to hopefully either report or establish populations of both species in the future. Therefore, the conclusion of our research is that Amblyomma americanum is the most prevalent tick species in southeastern Kansas, while Dermacentor variabilis ticks are also have a significant prevalence. That seasonality and life cycle has a greater impact on prevalence than temperature and humidity, which were relatively consistent compared to seasonality, where we saw either a large amount of adult and nymph ticks compared to larvae, and then in the later months of the summer, very high larva populations with low adult and nymph populations. And then Crawford County and Anderson County maps were updated with established and reported tick populations, and that follow-up surveillance in Anderson County and new surveillance in Montgomery, Labette, and Cherokee counties in the summer of 2024 is suggested, those being the circled red counties, which would increase reliable prevalence data and aid in public health efforts. And our references were the Center for Disease Control and Prevention, ticks of veterinary importance from the USD Agriculture Handbook, as well as acknowledgement that the research was funded by KDHE and correspondence was done by our professor. So Dr. Anurata Ghosh's email is listed. Thank you for listening to my presentation, and I hope this clearly and concisely portrayed the tick prevalence data that we were able to accumulate, as well as